Hello everyone. Today we are going to start with a new ERQ, that is the evaluate research on the role of neurotransmissions in human behavior. Now it says the command term is evaluate. It means we have to write the strengths and the weaknesses, as we all know, and I have told you many a times in my previous videos. The role of neurotransmission, means we have to choose 2 to 4 supporting studies from neurotransmission chapter in biological approach. So this is how I start my ERQ. I start by writing the introduction, and it goes like this. Neurotransmission is the process by which neurons transmit information and signals in order for certain bodily functions, either psychical or psychological, to occur. Communication between these neurons begins with an action potential, travels to the axon terminal of a presynaptic neuron. This electrical impulse causes vesicles to fuse with the membrane of the presynaptic neuron and releases certain molecules, neurotransmitters, which bind to the receptors on the postsynaptic neuron to carry on this action potential to the next neuron. These neurotransmitters are then released from the receptors and either return to the presynaptic neuron or are dissolved by enzymes. Often the role of neurotransmitters is studied indirectly. This is sometimes done by using an antagonist, that is a drug that blocks the receptor site on a neuron and does not allow a neurotransmitter to do its job. So here, as you can see, I have written an introduction, a very detailed one about the neurotransmission, what are the features and how does it work, and this is what you have to write. Post the introduction and the aim of the ERQ, that is 2 plus 2, 4 marks, you next write the supporting study, and this is what I write. A study by Troster showed that acetylcholine plays a role in the encoding of memories but not the retrieval of LTM. To carry out the study, there were 3 conditions. Each subject was injected with either a saline solution, a 5 or a 0.5 or a 0.8 mg solution of scopolamine, and acetylcholine antagonist. They then carried out 3 tests. In the first test, they were asked to recall a list of 14 words. Recall was tested immediately after reading the list and then after 45 minutes. The high scopolamine group recalled the least in both conditions. In the second test, participants were given a map of a fake state and asked to memorize the location of the cities. After 1 minute, they were given a blank map and a list of cities and asked to place them on the map. Once again, the high scopolamine group did poorly. Finally, participants were given a test of memories of famous people and events. They found no significant difference in the scores of the 3 conditions. It appears that acetylcholine may help a role in the encoding of memory, but not its retrieval. After this writing of a study, I write the evaluation part, that is the strength and the weaknesses, and this is what I write. The study is highly standardized, which allows other researchers to replicate the findings. The study is problematic in that scopolamine has strong side effects, so the researcher and the participant would know whether it was the placebo or not. The task is also very artificial and may not reflect how memories are usually created. Now I have write this, and next I am going to, after the evaluation, going to write my second supporting study. Antonova wanted to see if scopolamine affected activity in the hippocampus, particularly in the creation of spatial memories. The sample was made up of 20 healthy adult males. The study used a double-blind procedure with participants randomly allocated to one of 2 conditions, one in which participant would receive a scopolamine injection and the other would receive a placebo. At the beginning of the experiment, participants were put into an fMRI while playing a virtual reality game that was designed to test the participants' abilities to create spatial memories. The objective of the game was to navigate through an arena to reach a pole. Once they reached the pole, the screen would go blank for 30 seconds and the participants were told to rehearse how they got to the pole. Then they would appear in a different location in the arena to find the pole again. The fMRI measured the participants' brain activity. The researchers found that those who were injected with scopolamine demonstrated a significant reduction in the activation of the hippocampus when compared with the placebo group. This suggests that acetylcholine plays an important role in the encoding of spatial memories in humans. Although there was a higher rate of error in the scopolamine group, it wasn't a significant difference between the two. However, there was a significant difference in the activity of the hippocampus between the two groups. This implies that the design of the task itself was not ideal to show performance difference. And without the use of technology, there would be no way of knowing of the biological effects between the two groups. Now, as you know that I have written the two supporting studies, now I'm supposed to write the evaluation part, that is strengths and weaknesses of the second supporting study, and this is what I write. The study was a repeated measure design which allowed the researchers to eliminate participant variability. The study was also counterbalanced, with some doing the scopolamine condition first and some doing the placebo condition first to control for practice effect. In addition to that, the study was also a double-blind experiment, preventing researcher bias in the results. In order for the results to be considered reliable, the study would need to be replicated due to a small sample size. So here I'm done with my two supporting studies and evaluation of both the supporting studies. Now I write my last paragraph, that is the conclusion, which again carries 2 marks. Although these studies provide good insights into aspects of how neurotransmission affects behavior, which they cannot provide a full scope of the impact it has. Neurotransmission cannot be directly observed in the brain. So researchers use indirect measures such as animal research, fMRIs showing brain activity, or the use of antagonists. This means that the research is based on assumptions that may prove in the future not to be accurate. So here we are done with this ERQ, starting with the introduction, then aim of the study. Then I wrote one supporting study with its followed by an evaluation, then second supporting study followed by another evaluation, and then I conclude the study by writing a conclusion. So I hope you understood, and if you have any queries, write in the comments below. I'll be uploading more ERQs for you to refer.